Microbiology is the study of tiny living things. These organisms are called microbes or microorganisms. They are too small to see with our naked eyes alone. We need special tools and methods to study them. Plating techniques are fundamental methods in microbiology. They allow us to grow microbes in the laboratory. We can then see them and study their characteristics. This helps us understand their roles in health, disease, and the environment. Mastering these techniques is a key skill for any microbiologist. Plating involves spreading a sample containing microbes onto a nutrient medium. This medium is usually a gel-like substance called agar. The agar is held in a flat, round dish called a petri dish. The agar contains all the food that microbes need to grow. When a single microbe lands on the agar, it can multiply. It will form a visible cluster of cells. This cluster is known as a colony. Each colony often arises from a single microbial cell or a small group of identical cells. There are several different types of plating techniques. Each technique serves a specific purpose in the laboratory. For example, some methods help us to isolate pure cultures. A pure culture contains only one type of microbe. Other methods allow us to count the number of microbes in a sample. This is important for food safety or water quality testing. Understanding these different methods is crucial for accurate microbial analysis and research. These methods form the backbone of many microbiological investigations and discoveries. Learning plating techniques opens up a fascinating world. It allows us to visualize and manipulate organisms that are ordinarily invisible. These techniques require care, precision, and a good understanding of microbial life. With practice, anyone can become proficient in plating. This essay will explore various plating methods. We will discuss their purposes and applications. We will also cover important aspects like sterilization and incubation. These are vital for obtaining reliable and meaningful results in microbiology. The streak plate method is a very common technique. Its main purpose is to obtain isolated colonies of bacteria. These isolated colonies are essential for getting a pure culture. A pure culture contains only one species of microorganism. This is important because microbes usually live in mixed communities in nature. To study a specific microbe, we first need to separate it from others. The streak plate method is an efficient way to achieve this important separation. It is a foundational skill in microbiology laboratories worldwide. How is a streak plate performed? First, a small amount of microbial sample is taken. This is done using a sterile inoculating loop. The loop is then lightly streaked across one section of an agar plate. The plate is divided into 3 or 4 sections or quadrants. After streaking the first section, the loop is sterilized again. This is usually done by flaming it. Then the loop is used to drag some microbes from the first section into the second section. This process is repeated for the remaining sections, sterilizing the loop each time. The key idea behind streaking is dilution. As the loop is streaked across the agar surface, fewer and fewer microbial cells are deposited. In the initial sections, the growth will be very dense. Many cells will be close together. However, by the last sections, individual cells become well separated. When these separated cells grow and divide, they form distinct, isolated colonies. Each isolated colony is assumed to have grown from a single microbial cell. Therefore, all cells within that colony are genetically identical clones. Once isolated colonies are visible, a single colony can be picked. This single colony is then transferred to a fresh, sterile medium. This creates a pure culture. Pure cultures are necessary for many downstream applications. These include identifying the microbe, studying its metabolism, or testing its antibiotic susceptibility. The streak plate method, though simple in concept, requires practice. Good technique is essential for obtaining well-isolated colonies consistently. It remains a cornerstone of microbiological practice for isolating specific microorganisms. The spread plate method is another important plating technique. Its primary use is to determine the number of viable microbes in a sample. Viable microbes are those that are alive and able to grow and reproduce. This method allows us to quantify the concentration of these living cells. This is often expressed as colony-forming units per milliliter or per gram of sample. Knowing the number of viable bacteria is critical in many fields. This includes food microbiology, water quality analysis, and clinical diagnostics. To perform a spread plate, a small known volume of a diluted liquid sample is used. This sample is pipetted onto the center of a sterile agar plate. A special tool called a sterile spreader is then used. This spreader is often L-shaped and made of glass or plastic. The spreader is used to evenly distribute the liquid sample across the entire surface of the agar. It is important to spread the sample gently. This ensures that the agar surface is not damaged. The plate is usually rotated during spreading for even distribution. After spreading, the plates are incubated under appropriate conditions. During incubation, each viable microbial cell on the agar surface will grow. It will multiply to form a visible colony. Ideally, the dilution of the original sample should be such that the plate yields a countable number of colonies. This is typically between 30 and 300 colonies per plate. If there are too few colonies, the statistical accuracy is low. If there are too many colonies, they may merge and become difficult to count accurately. The spread plate method is preferred for organisms that are sensitive to heat. This is because the agar is already solidified when the sample is added. Unlike the pour plate method, the microbes are not mixed with molten agar. This method also ensures that all colonies grow on the surface of the agar. This can make them easier to see and pick for further study. Accurate pipetting and even spreading are crucial for obtaining reliable results with this technique. It is a widely used method for microbial enumeration. The pour plate method is also used for counting viable microorganisms in a sample. Like the spread plate method, it helps determine the number of colony-forming units. However, the procedure is quite different. In the pour plate technique, a known volume of the microbial sample, usually diluted, is first pipetted into an empty, sterile Petri dish. This is a key difference from the spread plate, where the sample goes onto solidified agar. This initial step sets the stage for a unique way of growing microbial colonies. Next, molten agar medium is added to the Petri dish containing the sample. The agar is usually kept in a water bath at around 45 to 50 degrees Celsius. This temperature is warm enough to keep the agar liquid, yet it is cool enough not to kill most of the microbes in the sample quickly. After adding the molten agar, the dish is gently swirled. This is done to mix the sample evenly with the agar. The agar is then allowed to solidify completely, trapping the microbes within the agar matrix. After solidification, the plates are inverted and incubated. During incubation, colonies will develop. A unique feature of the pour plate method is that colonies can grow both on the surface of the agar and within the agar itself. Subsurface colonies are often smaller and lens-shaped due to the physical constraint of the surrounding agar. Surface colonies will appear more like those seen on a streak or spread plate. Counting involves tallying both types of colonies to get the total colony-forming unit count. This method can sometimes lead to higher counts if microbes are present in clumps. One advantage of the pour plate method is that it can accommodate a larger volume of sample. For example, 1 milliliter, compared to the spread plate, typically 0.1 milliliters. This can be useful for samples with low microbial concentrations. However, some heat-sensitive organisms might be damaged by the temperature of the molten agar, even if it's brief. Also, retrieving colonies from within the agar for further study can be more challenging than picking surface colonies. It is another valuable tool for quantitative microbiology, especially for certain types of samples or organisms. Section 5. Why we must be clean. The critical role of sterilization. Sterilization is an absolutely essential concept in microbiology. It refers to the complete removal or destruction of all forms of microbial life. This includes bacteria, viruses, fungi, and even hardy bacterial spores. Without proper sterilization, our experiments and analyses would be meaningless. Contaminating microorganisms from the air, our hands, or unsterilized equipment could grow on our plates. This would lead to incorrect results and misinterpretations. Therefore, maintaining a sterile environment and using sterile materials is paramount. Several methods are used to achieve sterilization in a microbiology laboratory. Heat is one of the most common and effective methods. Autoclaving uses steam under high pressure, typically 121 degrees Celsius at 15 psi, to sterilize media, glassware, and other heat-stable items. Dry heat ovens, operating at higher temperatures like 160 to 170 degrees Celsius for longer periods, are used for items that cannot be exposed to steam, like some powders or oils. For inoculating loops, direct flaming until red hot ensures they are free of contaminants before use. Not all materials can withstand high temperatures. For heat-sensitive liquids, such as certain media components or antibiotic solutions, filtration is used. Special membrane filters with pore sizes small enough to trap bacteria, for example 0.22 micrometers, are employed. The liquid is passed through the filter, and the microbes are retained on the filter surface, yielding a sterile filtrate. Other methods include radiation, like ultraviolet light for surface sterilization, for instance, in biosafety cabinets, or ionizing radiation for sterilizing pre-packaged disposable labware like Petri dishes and pipettes. Aseptic technique is closely related to sterilization. It refers to the set of practices used to prevent contamination of sterile materials and cultures. This includes working near a Bunsen burner flame, which creates an upward current of sterile air. It also involves minimizing the exposure of sterile media to the air and handling sterile equipment carefully. Sterilization provides the sterile tools and media, while aseptic technique ensures they remain sterile during use. Both are fundamental for accurate and reliable work in microbiology. Failure in either can compromise entire experiments. Section 6. Giving microbes a home. The importance of incubation. Incubation is the process of providing specific environmental conditions. These conditions allow microbes on a plate to grow and multiply. After plating, Petri dishes are placed in an incubator. An incubator is a piece of equipment that maintains a controlled environment. The most critical factor controlled by an incubator is temperature. Different microorganisms have different optimal growth temperatures. For example, bacteria that commonly infect humans often grow best at 37 degrees Celsius, which is human body temperature. Other microbes might prefer cooler or warmer temperatures. The duration of incubation is also very important. Some bacteria can grow rapidly, forming visible colonies within 18 to 24 hours. Others, like some fungi or slow-growing bacteria, might require several days or even weeks of incubation. The incubation time must be sufficient for colonies to develop to a size where they can be easily seen and counted or isolated. However, over-incubation can lead to problems. Colonies might become too large and merge, or nutrients in the medium might become depleted, affecting microbial physiology. Besides temperature and time, the atmospheric composition can also be critical for incubation. Many bacteria, known as aerobes, require oxygen for growth. Standard incubators provide an air atmosphere. However, some bacteria are anaerobes. They cannot grow in the presence of oxygen, or oxygen is toxic to them. These organisms require special anaerobic incubators or anaerobic jars. These systems remove oxygen and often replace it with a mixture of other gases like nitrogen, hydrogen, and carbon dioxide. Other microbes, called microaerophiles, need low oxygen levels. Proper incubation ensures that the microbes we are interested in have the best chance to grow. It allows us to accurately assess their presence or numbers. Plates are usually incubated in an inverted position, meaning agar side up. This prevents condensation that might form on the lid from dripping onto the agar surface. Dripping condensation can cause colonies to spread and merge, making isolation or counting difficult. Careful control of all incubation parameters, temperature, time, and atmosphere is essential for obtaining reliable results from plating techniques. Section 7. Getting started. Essential tips for successful plating. For beginners learning microbiology plating techniques, a few practical tips can make a big difference. First and foremost is proper labeling. Always label your Petri dishes clearly before you start. Include essential information such as the date, sample identity, your initials, and any dilution factor. It's best to label the bottom of the dish, the part containing the agar, not the lid. Lids can be accidentally swapped, leading to confusion. Use a permanent marker for clear and lasting labels that won't smudge. Handling sterile materials correctly is crucial. When opening a sterile Petri dish, lift the lid only as much as necessary and keep it over the base to minimize airborne contamination. Work efficiently to reduce the time the agar surface is exposed. If using an inoculating loop, ensure it is properly sterilized by flaming until red hot before and after each use. Allow the loop to cool for a few seconds before touching a microbial culture or agar, as a hot loop can kill microbes or melt the agar surface. This cooling step is often overlooked by novices. Practice aseptic technique consistently. Work near a Bunsen burner flame if possible, as the flame creates a sterile field around your working area. Avoid talking, coughing, or unnecessary movements over open plates. Keep your lab bench clean and organized. When performing streak plates, be gentle. Do not gouge or tear the agar surface with the loop. A light touch is sufficient to transfer microbes. For spread plates, ensure the spreader is sterile and distribute the inoculum evenly for uniform colony distribution. Don't be discouraged by initial imperfect results. Plating techniques require practice to master. If your first streak plates don't yield well-isolated colonies, review your technique, and try again. Pay attention to details like the angle of the loop and the pattern of streaking. If using spread plates, ensure your dilutions are appropriate to get countable plates. Learning to troubleshoot common problems, such as contamination or confluent growth, is part of the learning process. Patience and persistence are key to becoming proficient in these fundamental skills. Section 8. What do you see? The power of accurate observation. After incubation, the next critical step is accurate observation of the plates. What you see on the agar plate provides a wealth of information. The first thing to note is the presence or absence of growth. If growth is present, you need to examine the colonies carefully. Colony morphology refers to the physical appearance of the colonies. This includes characteristics like size, shape, circular, irregular, filamentous, color, pigmented or non-pigmented, texture, smooth, rough, mucoid, elevation, flat, raised, convex, and margin, edge appearance, for example, entire, lobed. Observing these characteristics is important for several reasons. Different microbial species often produce colonies with distinct morphologies. This can provide initial clues for identifying the microorganisms present. For example, some bacteria produce vibrant pigments, while others form mucoid, slimy colonies. Variations in colony morphology within a supposedly pure culture might indicate contamination or dissociation of the culture. Careful observation is essential for distinguishing between different types of colonies on a mixed plate, especially when trying to isolate a specific microbe. When performing quantitative plating methods like spread plates or pour plates, accurate counting is vital. Count only those plates that have a statistically reliable number of colonies, typically between 30 and 300. Use a colony counter if available, which often has a magnifying glass and grid to aid counting. Record your counts meticulously. If you observe different types of colonies, you might need to count each type separately, depending on the purpose of your experiment. Be consistent in what you define as a colony. Beyond just counting and describing, it's important to interpret your observations in the context of your experiment. Are the results expected? Are there signs of contamination, like fungal colonies with a fuzzy appearance, or unexpected bacterial types? Did the controls work as they should? For example, a sterile media control should have no growth. Document all observations, including colony morphology and counts, in a lab notebook. Photographs can also be very useful for recording results. Accurate and detailed observation is the bridge between performing the technique and drawing meaningful conclusions. Section 9. Plating's impact, from lab bench to broader applications. Microbiology plating techniques are not just academic exercises confined to the teaching laboratory. They are indispensable tools with far-reaching applications in numerous fields. In clinical microbiology, plating is fundamental for diagnosing infectious diseases. Samples from patients, such as swabs, urine, or blood, are plated to isolate and identify pathogenic microorganisms. Once a pathogen is isolated, further tests like antibiotic susceptibility testing using Kirby-Bauer disc diffusion on agar plates can guide treatment decisions. This directly impacts patient care and outcomes. In food and water safety, plating techniques are used routinely. They help to monitor microbial contamination levels. For instance, pour plates or spread plates can determine the total bacterial count in a food product or a water sample. Specific selective and differential media can be used to detect the presence of particular pathogens, such as Salmonella or E. coli. These tests are crucial for ensuring that food is safe for consumption and water is safe for drinking. They help prevent outbreaks of foodborne and waterborne illnesses, protecting public health on a large scale. The pharmaceutical industry relies heavily on plating for various quality control processes. This includes sterility testing of injectable drugs and other medical products to ensure they are free from microbial contamination. Plating is also used in the development of new antibiotics, where researchers screen thousands of microbial cultures or synthetic compounds for antimicrobial activity. Environmental microbiology uses plating to study microbial communities in soil, water, and air, and to isolate microbes with unique metabolic capabilities, such as bioremediation, which is cleaning up pollutants. Furthermore, plating techniques are at the heart of basic microbiological research. They are essential for genetic studies, understanding microbial physiology, and exploring microbial diversity. Whether isolating a newly discovered bacterium, constructing genetically modified strains, or studying microbial interactions, plating provides the foundation for these investigations. The ability to grow, isolate, and quantify microbes on solid media, made possible by these simple yet powerful plating techniques, has revolutionized our understanding of the microbial world and continues to drive innovation across many scientific and industrial sectors.